Hello, this is Prash, and I'm going to give you a video abstract presentation on prioritizing SNPs and variants using regulatory systems genomics approach. The kind of question that we ask is, do traits penetrate into regulatory regions? And the challenge that we oversee is if there is any role of regulation or regulatory elements that are associated with these targeted genes. And we propose a kind of a 3-point classification scoring schema using these regulatory elements. So the problem here is, given pleiotropy and linkage disequilibrium, can we find causal SNPs associated with, say, a disease? And the disease of our interest here is clinical mastitis, which is associated in cattle, where the udder of the cow is swollen and it's a bacterial infection. The challenge that we encounter also is specific to the bioinformatics problem. We also ask if there is a heritable variation associated with these set of genes, and we also consider how many of these particular SNPs that we filter is heterozygous. And these are all the methods that we use. We also use some kind of statistics using Bayesian data integration. So in a genome browser such as Ensemble, if we key in clinical mastitis, we find these number of SNPs that are associated. The red means that these are the most significant number of features where the SNPs are underlying across these particular chromosomal loci. We propose a 3-point classification scoring schema. One is functional annotation, the constrained elements in the regulatory regions, each associated with different classification feature or a subfeature, like the presence of signal peptides in case of functional annotation, the constrained elements containing the evolutionarily conserved regions, whether or not if a SNP is conserved across all the eutherians or eukaryotes in this particular case, and the presence of enhancers and long non-coding RNAs associated with that particular SNP towards causality. So the conclusion here is annotation and curation is not the key. Interspersed with these regulatory elements or systems genomics approach, we need strong bioinformatics pipelines. We also look and ask if there are regulatory elements associated with these protein interactions. So commit next generation sequencing, we need to ask if the genome-wide association studies have really paved way for further regulatory genomics approach. And the most important thing is the DHS or the DNase hypersensitivity sites and the regulatory elements play a very important role towards deciphering the causality of a SNP for the disease. Thank you very much. If you have any questions, we look forward.